Good morning, guys. Wherever you are in this continent of ours, the channel smart underscore traveler memos presents biology practical paper which was done in 2002, and me being an active participant of marking that paper. I think you are with the right person to present that paper to you. So for all the candidates, wish you the best of luck as you go over what I'm going to present. I'm talking about candidates from all over the world because biology is universal. There's one time I said biology in Kenya is the same as biology in Uganda, the same as biology in Britain, is the same as biology in the United States of America. So, whatever you call something in biology is known universally. And that is why we have that topic known as classification. Anyway, let's go to the paper direct. Welcome, guys, and we see what we have gathered from there. Being presented the trophy of the year 1997 for biology. Okay. No. In this paper, question 1, you are provided with specimens labeled D1, D2, D3, and D4. And you are supposed to examine them, okay? There is something I would like to correct here. Confidentials are not at the end of the video, but they are in another video which you can always search from smart underscore memos. So let me make a correction there. So I've just made a correction there. So question 1, you are provided with specimens labeled D1, D2, D3, D4. So go to the video on confidentials and you see what the D1, D2, D3, D4 is and examine them. So we are supposed to draw and label D2, guys. I had to adjust my drawing. All right, you can see I'm still adjusting. So D1, draw and label. So it is drawn there. You can see we have the labels, the hook, persistent calyx. For hooks, reject spikes or spines or thorns. All right. And then we have the fruit. You can also call it pericarp. You can call it fruit wall, or you can call it pericarp. Epicarp, sorry. We have the accuracy mark. For this one, there should be no shading. Continuous outline all structures correctly drawn, and then proportionality. These ones are the ones we were looking at for you to get the accuracy mark. So, there are other writings there which can assist you. The fruit should be elongated. Persistent calyx should be proportional with the fruit size, and it should be at least 1/3 of the length of the fruit. Those are the writings in black. All right. So, persistent calyx, 1/3 of the fruit, at least 2, but not more than 4 persistent calyx. Hook face light direction. So the hook should face the right direction. Point it towards the fruit. The hooks should point towards the fruit. There is another one at the corner there. If D1 is interchanged with D2, mark the 2 and divide by 2. But mark the table in the better entry, because sometimes when putting the specimen, somebody might interchange. Choose if to use single line or double line, not both. All right. So those are the points which we looked at to award marks for specimen D1. Magnification lied between 3 and 5. And you have, of course, to put x3 or x5, or you say 3 times or 5 times. That would be okay. Draw and label specimen D2. For D2, the labels are papules or hairs, hair or papules of hair. You can put that, but you should reject hairy. Accept hair or papules of hair. All right. Fruit, epicarp, pericarp fruit wall. And for the accuracy mark again, continuous outline, all structures correctly drawn, proportionality, hairs 3 times longer than seeds. Excuse me. Hairs 3 times longer than fruit or seeds. Why? Dotted broken line arrow should not be awarded. At the corner there, the black tells you we are not going to accept dotted or broken line arrow. Fine. So there is a table there giving reasons, state the agent or method of dispersal of the specimens. So let me adjust this camera because the camera today is having issues. All right, so that you can see, guys. All right, I think that is good at there. So, specimen D1, agent of dispersal, animal, or you can say man, you get a mark. Reason is that presence of hooks or parachute-like. Specimen D2, weed, 1 mark. Presence of papules or hairs or parachute-like, or it is light. D3 is weed. Presence of wings, light, wing-like extensions of tester, paper-like, all right? And the D4, agent of method of dispersal is explosive mechanism or self-dispersal. And the reasons is that it has lines of weakness along the ovary wall. Or it has sutures, they are called sutures, these lines of weakness. Or you call them lines of dehiscence. State 3 the types of gynoecium and presentation of specimen D4. Type of gynoecium is monocarpous or monocarpal or monocarpella. Monocarpellary. Okay? Presentation is marginal presentation. Having gone through question 1, we go to question 2. You are provided with olive oil, liquids labeled R1 and R2, and an Irish potato. Label 2 test tubes A and B. Place 2 cubic centimeters of water into each test tube. Add 8 drops of olive oil into each test tube. To test tube A, add 8 drops of liquid R1. Okay, let me adjust this one again. All right. Shake both test tubes and allow to stand for 2 minutes. Record your observations. Test tube A. Oil broken up into smaller or tiny droplets or emulsified, which are dispersed, or you can say which are spread or which are suspended throughout, or you say milky, which are milky or which are cloudy. So oil broken up into tiny droplets which are cloudy. Or oil is emulsified. Reject white precipitate or white solution for being spread. Accept suspended on or in for floating of oil droplets. So you can say that. Observations for test tube B. Oil floats on water. You can say that. Or no mixing takes place. Okay? Or you say no 2 separate layers. Or you say immiscible. Immiscible. The liquids are immiscible. So, still recording your observations. Name the process that has taken place in test tube A. The process is known as emulsification. And this one is tied to test tube A. So if you fail the observations in test tube A, you don't get that mark. State the significance of the process named in A1 above to increase surface area. And again, it is tied to A1. All right? A2. So if you don't get the observations there, you cannot also not get this one. They are tied. So the moment the observation in test tube A is wrong, you don't get emulsification or increase surface area. Name the digestive juices in humans that has the same effect on oil as liquid L1. This one is bile. Reject other names. Take care of spelling. And it is tied to test tube A. So if you fail test tube A observations, my friend, you are also sunk here. You cannot get the mark. Region of the alimentary canal into which the juice is secreted is duodenum. And this one is tied to 4. So if you fail bile, then you don't get that mark. Label 2 test tube C and D. This is B. Place 2 cubic centimeters of liquid L2 into each and a drop of iodine in each test tube. Record your observation, and you find that you can get the following colors: blue, black, black, dark blue, blue, grayish black, bluish purple, or purple, blue, purple, purplish blue. All these colors could have resulted from the way that liquid was prepared. Okay, but they are all correct. Suggest the identity of L2. L2 is nothing but starch because of the blue-black result. Cut a cube whose sides are 1 centimeter from the Irish potato provided. Crush the cube to obtain a paste. Place the paste in test tube labeled C. Leave the setup for at least 30 minutes. Record your observation after 30 minutes, and you find that contents of D remains unchanged, or you say content of D becomes colorless, as in one above. All right. C disappears. Turned yellow, orange, brown, purple, pink, light yellow, land, brick land, turned to B1. D is the control. Reject purple if purple in B1. So you don't mark purple here if the answer given to B1 was purple. Account for the results in B3 above. So, enzyme amylase in potato breaks down starch, hydrolyzes, converts starch, all those ones, into simple sugars, or into maltose, or into glucose. You can mention anything there, like those are the ones that do not give blue-black color with iodine, or you say absence of starch. So the sentence should read like this, because we said in paper 3 or any examination, you cannot give answers with a stroke. So it should read this way. Enzyme amylase in potato breaks down starch, all right, into simple sugars that do not give blue-black color with iodine. Cut out another cube whose sides are 1 centimeter from the Irish potato. Crush the cube. Place the crushed paste into a test tube. Carry out food test with the reagent provided. Procedure. Add equal amounts of Benedict solution to the paste and boil, or you heat, or you warm, except water bath. That is, you put them in a water bath. Add equal amount of Benedict solution to the paste and put them in a water bath. So you can also say that. Result, green, okay, or yellow, or orange, or brown, or brick red, depending with the heating, how you heated. So reject red. It must be brick red. So any of those colors can be got depending on how you heated. Account for the result in C1 above. Starch in potato is converted to maltose or simple sugar and so forth, or reducing sugar by enzymes or by amylase or by maltase. So in a sentence, you can say, starch in potato is converted to simple sugar by amylase, and you see I've not put strokes, so you have to be very careful. Never put strokes. Reject styrene for the enzyme and also reject diastase. It must be maltase or amylase. Finally, we have question 3. You are provided with specimen labeled Q and R. Examine them. Giving reasons, state the phylum of the specimens. Phylum, Arthropoda. Reject spelling, but mark the reasons. So, giving reason, the phylum of the specimen, the phylum is Arthropoda. Reject wrong spelling, but mark reasons. Reasons, they are tied to the correct phylum. All right? Segmented body, exoskeleton, or you can say chitin or ones to that effect. Jointed appendages, or jointed legs or jointed ribs is accepted, but you reject joint appendages if the one is not jointed. If you say joint, you are not going to get it. Name the class to which the specimens belong. Insecta. Reject wrong spelling, but mark reasons. Insecta. So you cannot say insect. State the features common to both specimens that are characteristic of the class mentioned. So 3 body parts, 3 pairs of legs or 6 legs, or a pair of antennae, or open. This is an open question. Mark any correct 3. But note that B2 is tied to B1. So if you don't get the class, you cannot get the characteristics. State the differences between Q and R, 5 marks. So now we give the differences there. The first one is for Q, you have hard body versus soft body or exoskeleton. All right. All right. Hey. Something went wrong with the camera. And therefore, let's look at the differences again now for Q and R. So hand, okay, was a soft. Reject specific body parts. Winged, the other one is wingless. Reject incorrect number of wings. Head with snout. Head with mandibles. Snout is sometimes known as rostrum. Antennae are swollen. Antennae are straight. All right, and so on. Has eyes. The other one has no eyes, but reject the one bride. You cannot say the other one is bride. Other than feeding, how are the mouth parts of specimen Q and R adapting their function? Snout for boring into grains. Okay? And the other ones, of course, without putting strokes. Then we have specimen R, enlarged scissor-like mandibles for defense. Reject pointed mandibles. And note that the feature is tied to the adaptation. State the difference and functions of the wings of Q. Difference: outer wings, hand, while inner wings are soft. Outer wings are opaque, while inner wings are translucent. Okay, you just have to match the comparisons. Functions: outer wings for protection, inner wings for flight. Or here is a table. You can make a table for comparison. Outer wing, forewing, inner wing. So we have thick, thin. Hand, soft. Opaque, translucent. Stiff, flexible. So remove one leg from specimen R, draw and label it. All right? So we have removed the leg, although, okay, the camera has just fallen down, the leg has been removed. Okay, camera has fallen down. So now we are labeling it, and you can see A can be closed or open. That was not A, sorry. That arrow there shows that part which can be closed or open. Then we have the femur, we have the tibia, we have the tarsals, and we have the claw. For the accuracy mark again, we go closer and turn this thing like that. And you can see for the accuracy mark, we have continuous outline, femur crossed at base, tarsals at least 3 and not more than 4, proportionality, all right, femur, and femur, femur wider than others, femur wider than others, then all structures and parts correctly drawn, all joints to be shown, spikes if present or absent does not spoil. So all joints to be shown, all those points there, continuous outline, femur cross, and so forth, are for the accuracy mark. All right. You are giving 4 marks. You have got 4 marks, maximum 3, plus the accuracy mark. Then for the label, consider word spellings. Label marks check parts that open and into each other for you to give the rebel marks. All right, you check whether the parts are opening to each other for you to award that mark. So, of course, it's problematic. That's why you see now the way I'm placing the camera for me to get the full view of the diagram. So guys, that's the way you draw. That's the diagram now, very clear with the rebels, femur, tibia. All right, the camera is shaking. Femur, tibia, tarsus, claw, and then you can see that part which is supposed to be open. So this is after you remove one leg from specimen so that you draw and label it, where you are getting 4 marks, but maximum label is 3. All right, so that you give the accuracy mark and the total will be 4. So that one has been problematic, guys. Okay, that part. So I'll just end the video by thanking all those people who helped me come up with this video. May God bless you, and then ask you people to continue subscribing to this channel for more videos like this. So guys, goodbye and see you for 2 or 3 practical. I hope if you find any problems in this one, you give the comments so that we can try to improve. If there is an area you don't see, of course, guys, we encourage you to give comments so that we can give encouraging so that your comments can make me improve the videos so that you can also see the videos very, very well, guys. So, goodbye and wait for the next video.